It's all yours, SFT.
Thanks so much, Praise. Thanks so much, SpeedOfSound. Guys, this is Standing for Truth tonight. We got a fun broadcast. It's going to be more so an informal discussion, so I'm looking forward to it. Thanks again for joining me in this discussion, SpeedOfSound. You are a guest tonight, so we're going to keep this profitable, respectful, just a good back and forth dialogue. What I want to do, though, is give us both a couple brief just introductions, kind of. If we start with you, tell us where you're coming from, who you are, what your goal in this battle is, I guess. Make a brief case. I think we're focusing more on genetics. What's more consistent, creation or evolution in light of genetics? So I'm going to hand it over to you. Take as much time as you need. I'll just take roughly the same time that you take, and then we'll just go back and forth for a bit.
Okay. Let me know if there's any audio problems, because I'm not sure about this mic. Let's see. I'm a hard atheist, strict naturalist. I see everything happening through naturalism, and I try to explain things from that perspective. And I see I have great issues with your group and with creationists. The problem I see is that you don't really try to find out how things work naturalistically. And there's an assumption of a worldview that sort of intervenes. And I like to dig a little bit deeper than that. But the odd thing about it is that I have a naturalistic worldview, but I do the same thing. I'm very skeptical of evolution and of every piece of evolution that I read. So when I study evolution, I noticed this about 6 months ago. I'm more skeptical about what I'm reading than you guys are, I think. I don't believe anything until I see it explained. There's music. So anyway, the naturalistic case requires that you have doubt about every possible mechanism because you don't know how things happened. So you always have to doubt what you're finding. And I think that's kind of what scientists do. Now, you guys talk about evolutionists and evolution as a religion. There's a whole lot of background noise somewhere. Anyway, I sort of like calling me an evolutionist is kind of like swearing at me. I assume you don't mind being called a creationist. But I do mind being called an evolutionist because I approach it with a lot of skepticism. I have no options, though, because I do not have a belief in any sort of supernatural entity or entity outside of this universe that could have created things. So I am kind of stuck with finding a naturalistic explanation. And most of all, I have problems with some of the research you've done, some of the things you've said. I think some of the things I'd like to cover is genetic entropy, ApoA1. We could lead with that one, and some of the other approaches that you take. I find that you are, in my opinion, putting out a lot of misinformation, and I've checked on quite a bit of it, and it concerns me. So maybe we can both learn something here tonight if we get into it, but that's about all I got. I would like to talk about ApoA1, Milano. I've been reading just recently about that. So that's the start. So what do you think of ApoA1 and all of that? I'm good.
Okay, awesome. Thank you so much for that. I'll cover a couple things first, just to kind of go over my position, and then we can certainly, if you want to focus on, I believe you said the melanoprotein and genetic entropy, so we can certainly lean in with those things. You've mentioned that you're skeptical, which is good, and since we're going to be talking about genetic entropy, I mean, I do believe that it's quite evident, and the fact that you're skeptical, I'm hoping that I can maybe show you the validity of it, because it's not just creationists like myself that agree that mankind is degenerating and accumulating these deleterious mutations, but it's also leading human population geneticists as well, and they certainly agree that, you know, we're accumulating these deleterious mutations, and the problem is we're accumulating them faster than they're actually being selected away. So what that indicates is that the human genome is degenerating. If these mutations that are unselectable, that are essentially invisible to natural selection, there needs to be a type of selection that can select away such mutations without them building up in the genome and in the population as a whole. Because I mean, it's pretty well established that man accumulates about 100 new mutations per person per generation. Sometimes more, sometimes less. This is roughly the average. At every cell division, we accumulate 3 new mutations, and primarily that's why we die. But those are in the somatic cells. So within the reproductive cells, when you've got mutations and they're passed on, natural selection at the end of the day is only going to rid the population of the most detrimental and deleterious mutations and amplify the best beneficial mutations. So sure, we can discuss a few beneficial mutations. A lot of them now are known to be based more on epigenetic regulation. Melanoprotein, for example, whether it is truly beneficial, whether it can actually counterbalance the accumulating damage, doesn't really matter, because the bigger picture is the fact that beneficial mutations are rare. Therefore, even if I give, say, the evolutionist 1 or 2 or maybe 5 truly beneficial mutations, 1 or 2 or 5 beneficial mutations are not going to counterbalance the damage based on the accumulating mutations. So the evolutionists would have to present us with, you know, thousands of truly beneficial mutations that can counterbalance the information loss. So those are the 2 things you want to talk about first. So that's cool. We'll get into that. First, I want to say, though, just kind of more my position, because you stated you're skeptical, you're open to the evidence, and you were stating that, you know, we support our position based on an assumption or a worldview. So if I do take that assumption, okay, that the Bible, and more specifically Genesis, is the history book of the universe as it claims to be, with that basic assumption, as you stated that I have, and I have no problem with, we can actually make predictions, retrodictions. So I find that, you know, those that are anti-Bible, anti-Genesis, for example, and I can certainly just call you anti-Bible or skeptical. I know you don't like the term evolutionist, but they seem to constantly attack the Bible and scoff at a literal Adam and Eve, for example. But I find that those that are skeptical really need to be asking themselves this question if they're truly seeking an answer regarding Adam and Eve in the Bible. And I'd like you to answer this too when we get going, but what would they honestly expect if Adam and Eve were true? What are the true expectations? And I find evolutionists approach this question through conversations with them, interacting with them. They always approach it from an evolutionary worldview, because we all have worldviews. That's the thing. A lot of it comes down to a worldview issue. And I find that this hinders them from assessing the actual data and evidence, and therefore coming to an appropriate conclusion. So I would say they need to address the real question, and tonight you would have to address the real question. Because if Adam and Eve were true, which is what I'm proposing, what should we expect? Well, we should obviously expect one female ancestor of all people on the planet. That's obvious. And the fact is, that's actually true. There should also be only one male ancestor of all people on the planet. Turns out that's also true. Genetic data confirms these expectations. You and I confirm those expectations. A biblical prediction is one male and one female. And this does not have to be true if evolution is true, but it has to be true if the Bible is true. And incredible data that we've discovered involves mitochondrial DNA, Y chromosome DNA, the Y chromosome, for example. And just to go over the Y chromosome, for example, we know that that's passed on unbroken from father to son. Every once in a while, a mistake happens, as we were talking about earlier, mutations accumulate. But every time this happens, a new branch in the family tree is formed. If we look at all the branches in the world, they go back to a single person, not a chimpanzee, but to a man. And based on observed mutations, the observed mutation rate, this man lived just thousands of years ago. That's Y chromosome Adam. And I can go on and on and on about mitochondrial Eve and mitochondrial DNA. It actually turns out to be exactly the same. We see low genetic diversity. That's why evolutionists have to resort to a population bottleneck, which is more like an ad hoc, post hoc embellishment. So we can get into that as well. But I'm glad that you're skeptical. I understand we both have worldviews, but I think if you really, really pay attention to what you would expect if our worldview was true, you'd find that the scientific data fits perfectly with that basic assumption. So I'm good there. That's all I got to say kind of on the spot. But if you wanted to lead in, do you want me to go refer to you as Mike or Speed of Sound?
Speed of Sound is fine.
Okay.
Better than Mike.
What are your thoughts on that? And
that opens up a whole lot of discussion that we can handle a little bit later. First thing I wanted to talk about is ApoA1 Milano. Now, I read a bunch of books, and I read a bit by Raw Matt on ApoA1, and he claims that and you've claimed that it's epigenetic, that it's deleterious, that it's lack of specificity, I think is what your term is. And none of these things apply to ApoA1 Milano. That is a really good example of a beneficial mutation, which is purely a mutation. And Raw Matt had said it was something to do with epigenetics, or that it was set off by people being sick or something like that, but none of it made any sense at all. ApoA1 is a mutation. It's an arginine that was replaced by a cysteine. And that will cause the proteins, okay, ApoA1 automatically always has dimerized, trimerized, tetramerized, and even formed octamers. That's eight molecules together, 4 molecules together, 2 and, whatever, 3. The ApoA1 Milano causes it to only dimerize and tetramerize. That means 2 or 4 of them get together. And it seems to change the shape of the molecule just a little bit so that it's highly, highly more effective at mopping up cholesterol that's excreted from the cells. It has nothing to do with epigenetics. It's a true mutation. It's not something that you're going to see infiltrate the entire population. It's not going to drive evolution. It's primarily going to affect people that are beyond the reproductive stage in their life anyway. Limiting heart disease. I have cats fighting in the background. Stop it.
Okay.
Anyway, so all of the things that you guys have said about ApoA1 Milano do not make any sense. And then to make matters worse, in Matt's book, he goes ahead and he says that the specificity already existed in the wild type A1 protein before the mutation occurred. And then he refers to a paper right here. And this paper isn't about anything like that. This paper is about what happens to ApoA1 when it's inside the cell. It has a pre-peptide attached to it, and it gets cleaved. And this paper talks about how we don't know why that is, and we don't know much about it. And so your references don't even match up with what you're talking about. And that is my problem with your channel. I mean, you guys say things, you do not put links to the primary sources. RJ Downard talks about primary sources. And you need to do that. You need to link the papers so that people who do care to go out and read them can go out and read them. And you've said many times that these papers are very technical. Well, that's what I do. I read technical papers. So, and I have a whole stack of books here on biology and molecular biology and population genetics. And I go over these things. But anyway, that's my thing on ApoA1. But even with that, you've discounted it and said, well, hell, we'll give you that one. We'll give you another one. We'll give you 5, but you got to come up with millions of them. And that leads us to the broader discussion of evolution and whether or not we need to discover millions of beneficial mutations like this one in order to believe in evolution. That isn't why anyone believes that evolution happens, because they found 1 or 2 cases like ApoA1. So anyway, I think you need to go over your literature on ApoA1, rewrite it, or I'll stop talking about it entirely, because it doesn't help your case at all. Other than that, you have to concede that it is a beneficial mutation that occurred. So.
Okay, so before I get a little more into detail on that, would you agree that the protein structure regarding the Milano example already existed prior to the advantage seen?
Well, the protein structure as it was before the cysteine got there, yes. But with the cysteine there, when 2 molecules attach together with a cysteine bond, what could happen is that molecule could be bent just very slightly. Here's the protein. You know, they all fold, every one of them, basically.
Okay, so
pentapeptide.
So are you disagreeing with the claim that it worked as a broad spectrum antioxidant, which now, because of escape through adaptive conflict, which does and is known to happen, which isn't going to answer the question of net gain versus net loss, which is what it comes down to regarding genetic entropy. But if it only functions to clean up, say, LDL cholesterol, right, low density lipoproteins, going from a multi-function to a single function just to keep the organism alive, which is exactly what's happening in this example. It's just organismal adaptation through environmental conditions. So if that's exactly what we see, going from a multi-function to single function for adaptive purposes, then how is that going to answer the question of net gain versus net loss? And secondly, how is that truly a beneficial mutation that is not deleterious in any way? Because you said it's not a loss in specificity, that it's actually truly a gain in novel information. And that's not even the point. It's just, can it counterbalance the damage, right? Because we're saying for many reasons that we can get into, beneficial mutations, which I hope you agree with me on the fact that they're rare, we're saying they cannot sufficiently compensate for the relentless influx of deleterious mutations. So if this is just general adaptation, whether it's epigenetic regulation or it's truly a mutation, it still did go from a multifunction to a single function. So how's that going to answer the bigger picture, which is what you mentioned? And that would be the bigger picture, right? Did an amoeba-like organism or a bacteria-like organism evolve over time incrementally into a whale and all the other life that we have? Because that's not going to happen through loss of information and genetic degeneration. So is that all it is? Is organismal genetic adaptation, whether it's epigenetic or mutation? The data does say it's a loss of specificity. So how are you going to use that to address the bigger picture? Go ahead.
Okay. You're running back and forth across the football field with the goalposts. We can talk about ApoA1 Milano, that's that specific thing, but you can't keep going to this net gain, net loss, and genetic entropy. We'll get to that later. Let's just talk about this thing first. First of all, it doesn't absorb low-density lipoproteins. High-density lipoproteins help get rid of cholesterol in the system. And it has several other functions, immunological functions. It has functions in the brain for cleaning out the brain, helping with Alzheimer's. And it has an antioxidant property. Basically, that chylomicron that is built out of this HDL has the capacity to work as an antioxidant because there are peroxides, and those are the things that damage your arteries. There's nothing about this thing. There's nothing in the research that indicates it has lost any function at all. It has only gained function. That's the only thing that they found so far. That's why so many pharmaceutical companies dumped hundreds of millions of dollars in to try to develop a drug to do this. They were not able to. The only way they could make mice better is to give them IV APO-A1 Milano. And it actually reverses hardening of the arteries. So this is a magical mutation. There is nothing wrong with this mutation. It has nothing to do with lack of specificity or anything. It still functions in all those other capacities. The only thing it doesn't do is it doesn't form oddball tetramers and octamers anymore. It forms two kinds of collections, and they seem to work much better than the other ones. So this one, like I say, you need to go back and read the papers that have been written on this, or look at the structure and how it works. It's clear that you don't know how it works exactly, and I don't expect you to be a biochemist, but what you said didn't make any sense. So, and it's not epigenetic. It has nothing to do with adaptation at all.
Are you disagreeing with the fact that this protein that we're referring to here has lost a lot of activity for making the HDLs that you're talking about? Wouldn't you agree that making HDLs is important? I mean, you just covered that.
That's what it does. It is an HDL. It's a high density lipoprotein.
Right.
It makes a chylomicron, and it does it just as well. In fact, there's nothing.
1 second, just because I don't want to lose my. Because you mentioned a cysteine. You mentioned that it was a single amino acid, and
arginine to cysteine.
And then there was a replacement observed with a cysteine residue, but that protein essentially assembles HDLs, and those are involved in removing LDL.
Cholesterol.
Yeah. Exactly. So that if it's worse at doing its job, that would be a loss of specificity.
It's much better at doing its job. That's the whole point. That's why everybody's excited about it, because it's far better at doing its job. It was a wonderful mutation that everybody should have. I wish I had it. I'd live for another, 30, 40 years. There is nothing in the literature that indicates that there's anything wrong with this protein. And also, Sanford and a bunch of others.
If it's lost some activity for producing HDLs, which is a good thing.
No, it hasn't.
Okay, so your disagreement. Okay, so your disagreement would be that it's not truly a loss of specificity. It's not truly epigenetic or environmental related, but that it's truly something beneficial. That's a mutation that has no deleterious effects. Okay, but let's go to the bigger picture. Okay, is this the type of example of adaptive gain that you believe is going to solve this problem of genetic degeneration? I mean, do you agree that we do accumulate nearly 100 new mutations per person per generation as a human being? I mean, do you disagree with that?
I think it's probably more like 60, and most of them are in the, I don't like to call it the junkyard, but the vital yard. Basically that 88% of the genome that is not vital to function. There's or not the vital yard, but it's basically the warehouse, and that warehouse is where evolution happens. That warehouse of all those mRNAs that are being translated that they found in the ENCODE project. Basically, that's the best news for evolution that we've ever had, that these things are being partially translated. Where the hell was I going with that? I lost my train of thought on that.
So are you referring to the ENCODE project that revealed that over 80% of the genome is transcribed into RNA? You're saying that was an important
transcribed at a very low rate. Yes.
Well, it's definitely it's definitely biochemical. So in other words, it suggests function. It gives us a trajectory to look to more experiments, let's say, gene knockout tests, for example, that are really the best way to discover whether or not, a specific gene or gene sequence is functional. But you would agree, so you would agree that ENCODE did at least reveal that over 80% of the genome is actively transcribed in RNA, but you said that it's transcribed at a low level, is what I think I heard you say?
Very low levels, yes. And none of them are vital that we know of. Or there are going to be a few that they find that are vital. I mean, they're discovering new genes every day and figuring out how useful they are. Most of them are transcribed. They do have an effect on the overall biochemistry of the cell, but the effect is very small. If it was not, all of the mutations, those 100 mutations you talk about, if they were not, they would kill us outright. They would kill our species off in long ago, basically.
Are you saying that the mutations that would occur, say in some of the non-coding regions, if it's on a gene that's transcribed at a low level, you're saying that it would only have a tiny effect on fitness?
Very tiny. It probably wouldn't affect fitness at all.
No,
It'd
that's
be
the point.
Neutral.
That's the point. Is most mutations are nearly neutral. There's a very subtle effect on fitness. It's kind of like a single spelling mistake in a book the size of an encyclopedia, right? It's going to be very subtle, but the buildup and the accumulation of those types of mutations over time, that's what's degenerating our information systems. And we can see this. We can see entropic degeneration on all levels. I mean, you mentioned Alzheimer's earlier. I mean, cancers are skyrocketing. Autism clearly has a genetic component to it. So I would just simply ask you, based on the fact that these mutations that are nearly neutral only have a very trivial effect on fitness, okay? 100 new mutations per person per generation. If natural selection is only acting on the most detrimental of mutations and amplifying only the best, let's say the melano one, for example, let's say it did amplify that one. Okay. What type of selection, though, is going to select away the near neutral mutations that accumulate relentlessly? That is the bigger picture that you yourself even talked about. So what type of selection could do such a thing?
That brings us to the bigger problem. What do you think GCTA, when it rusts, becomes? The genetic code, GCTA, you talk about the rusty car analogy and the book analogy where we're replacing things. The book analogy is better, but the rusty car analogy, what does rusty GCTA look like to you? What do you think it is?
So
it's still GCTA.
Our DNA code, of course, ATCG. If there is a mutation, let's say a point mutation on a single nucleotide, okay, if it damages it and leads to disease or has a tiny fitness effect, or if it's a deletion, the rust on a car, right, it accumulates relentlessly. The analogy, and no analogy is perfect, is because rust on a car, it generally builds up unnoticed until it reaches a point where no matter if you're changing the tires, changing the brakes, getting a new windshield, getting an oil change, the car will ultimately degenerate. Because although you're replacing some of the broken parts, let's say wear and tear items like brakes, it doesn't matter because the car is still degenerating in many other ways. You can't stop.
Not degenerating.
Are you saying that you can keep a car going for 20, 30, 40 years?
Not cars. That's why it's a bad analogy. The only way a car analogy would work is if, say your tires rotated themselves. That was a mutation. Your taillights switch places, a mutation. One mutation can be extremely deleterious. It could be fatal. Suppose all of a sudden one mutation, your engine jumps into the back seat. Not going to work, right? I mean, there are things. The natural selection. Okay, Kimura. I got his books here, basically.
Yeah.
I don't know where, but I have his books. Kimura talked about neutral mutation as being basically that warehouse of random mutations that were turning around, not being selected for, not killing us, just out there. And those are the things that drive evolution. And that was Kimura's entire point. He was saying that mutation, he was talking about mutation-driven evolution. And I sort of agree with him. We've got so much to talk about. The rusty car thing doesn't work because GCTA, when it rusts, is still GCTA. I mean, it's still a genetic code. It's still biologically active.
Right, but
it
even
does something. But if it does something,
mutation or deletion can lead to disease.
It can lead to disease like Alzheimer's, you mentioned, and cancer, which don't have a damn thing to do with evolution. You are long over your reproductive range by the time you get Alzheimer's.
Right, but the point is the evolutionist is trying to tell us that a bacteria-like organism turned into a whale over time through incremental changes. Okay, there's a lot of information that needs to be added. Okay, but if all we see is loss of it, I mean, there are many lines of evidence that supports this genetic degeneration that I'm talking about, and the whole point is that would strongly indicate that this ape to man type evolution is literally impossible. And the reason why is because the direction of change. Okay, I'm sure we both agree that evolution, at least biological evolution, means a change in allele frequency in populations over time. But what we see, the degradation of our genome, is that the direction of change is downward, never upward.
You're conflating stuff. Okay, you're familiar with this book, right? This is Alexei Kondrashov.
Yes, he had a paper. Why are we not extinct 100 times over? Because like you said, these populations.
A facetious title for his paper, which you took and you spread as if he was saying we should be dead 100 times. He goes on in the paper to explain why we're not. Right. And in this book, he explains why we're not. But we're conflating two things. We're conflating evolution, which is the broad picture, with humanity. And humans right now, we have 9 billion of us almost, and we have a lot of mutations, and we always have had, and they're going to keep popping up like hemophilia and whatever. And these mutations there's probably going to be new diseases popping up and old ones dying away. They're going to genetically drift out of the population. It doesn't matter. The only thing that matters, 50%, in my estimate, of fertilized gametes die in the zygotic stage, and you don't even know you're pregnant. God is the biggest aborter of all. There must be zygotes up the ass in heaven. Because you die if you get if you get a mutation that is vitally on a gene that's vitally important. That's why you see so little mutation in vital genes. You see it all in that big junkyard.
Yeah, but that's just support for my position. Based on our thesis,
mine too.
Just 1 second. I don't want to get too far because you brought up Kimura earlier. Okay, so the crucial issue here, is that most beneficial mutations are too subtle to be selected for by natural selection. Now, Kimura and what he talked about, is he would agree that the crucial issue then is about defining the correct distribution of mutation effects. I want to ask you, does he show the beneficial distribution in his papers, in his books, or does he kind of just say it without any actual evidence that yes, beneficial mutations will truly counterbalance, the damage from the accumulating near neutral mutations? Because you even admitted earlier that these neutral mutations do have a very subtle effect on fitness. So that definitely doesn't help your case. So do you know if Kimura did define the correct distribution of mutation effects?
Kimura's primary thing was to tell us that there is this whole section on that graph of neutral mutations that accumulate, and they drive evolution. That they're sort of the workshop for evolution that are happening behind the scenes. And that was basically his point. The neutral ones, yes. And neutral is kind of, you have to be careful with neutral.
Right. I like to say near neutral. Are those neutral mutations driving evolution because they are building up and not being selected away?
Not being selected away, and they're not doing any damage. When they do damage.
And that's the problem.
They can do one or two things.
They are doing damage.
They can do another thing too.
Very, very subtle effects. Here's the thing is, I think it's pretty obvious that because you just agreed that most mutations are nearly neutral. That means they fall within the no selection zone, okay? But they're all very slightly deleterious. So natural selection, like we agreed earlier, yeah, it's going to amplify some of the best beneficials, like melano, for example, okay? Even if I gave you that, even if I gave you another one, that doesn't necessarily help the case because the entire zone is still undergoing degeneration. I mean, you really do need to answer the question, what type of selection is going to select away the nearly neutral mutations that are accumulating in the genome? Like I said, I mean, I know you don't like the rusty car analogy, but a single spelling mistake in a book the size of an encyclopedia, yeah, that one single spelling mistake, okay, akin to say a mutation on a single nucleotide, won't have an overall effect on the book. But over time, it's the buildup of them. And if nobody's selecting those away, well, all you're gonna see is further degeneration. So can you spend some time answering that question?
I can. Population genetics, it's well known that when there are beneficial mutations, even though they aren't that common, those beneficial mutations under certain environmental influences take off. They take off and they spread throughout the population. And they spread out through the population, not to the point of 100%, but they very quick, well, they can if there's a bottleneck or there's something that happens in the environment, but they spread very, very quickly up to about 90%. It happens very fast. And that's what, and Camaro made that point. And the neutral mutations, they do nothing at all. They don't affect reproduction. They do not affect the fact that that our race is going on. Humans may be screwed because we keep everything. We keep every mutation, and we're getting quite large, and we may develop some really interesting genetic problems. And Kondrashov was worried about that for humans, not for insects, not for bacteria, not for chimpanzees. It's not a problem. This background, these near neutral mutations are just proteins mutating, changing, trying it out. Each one has its own unique shape. All proteins fold, even the bad ones, okay? If they don't kill you and they don't affect your reproductive rate, okay, they will end up to some extent being in the population if they don't drift out. Right. Every now and then, one of those will turn out to be beneficial, like ApoA1 melano. And if it's beneficial in such a way that it allows that species to survive some sort of environmental catastrophe, then you have evolution, and that's how it happens.
Would you agree, though? Okay, I can see that our disagreement, obviously, before I ask you this question, is whether or not these mutations are truly neutral or nearly neutral. I believe that, you know, what we now know about the genome as a whole, the non-coding regions versus the coding regions, that these neutral mutations, especially if they occur in, let's say, a non-coding region, which we now know is highly functional, it's going to be nearly neutral. It's going to have a small effect on fitness. But would you agree that selection itself happens on the level of the organism, on the whole organism, right? Is it acting on the phenotype or is it acting on the genotype?
It acts on the whole organism, on the phenotype.
Right. So these neutral mutations, okay, I do remember you at least saying that they're going to be at least subtle, very, very tiny effects on fitness. There's no way that it can't have any type of effect on genotype, okay? So if selection is only acting on the level of the whole organism, okay, it's only acting on the phenotype, how is it going to stop that loss of information, okay, due to the low impact mutation accumulation, which, as you agree, I'm sure, is happening on the molecular level? So I think you would have to just imagine that these mutations, these 100 new mutations per person per generation, are truly neutral, no negative effect at all, when I think that can be easily disproven. So if selection is amplifying the best beneficials and getting rid of the worst detrimental mutations, you do have to spend some time telling us or giving us a type of selection that is going to be able to select away these low impact, near neutral deleterious mutations.
It doesn't have to select them away. 99% of them don't do anything at all that we care about. Okay, they may do something that a 99-year-old man will care about. You know, he gets arthritis a little bit early or whatever. But it probably doesn't have a lot to do with that either. Most of it happens in that huge, vast area that is not vital to the organism. Okay, and it's going to affect the biochemistry. Now, I disagree with a lot of geneticists in that I think there are more beneficial mutations, or than a higher fraction than they believe, that there are a lot more of them out there that basically do something. When you're in that neutral area, it doesn't matter. You're on both sides of that line. You're beneficial, negative, right? You're affecting the biochemistry of the cell, but for the most part, the cell doesn't care because the genes that are vital are clean. They cannot be mutated unless it kills you, and those genes are 99% of your metabolism. The 1% of your metabolism that you're worried about, the 1% that humans are worried about when they get ill or they get stomach aches or they get whatever, depression or all of that, have nothing to do with evolution. So we have to quit. If you want to talk about evolution, we have to talk about the world without men, basically, because we're conflating, we're bringing a whole lot of garbage in that doesn't belong in the discussion of evolution. So, but those beneficial mutations will appear out of that mass. You see, you're stuck on this rusty idea that there's something going to hell and it's getting worse every time. It doesn't. Those 100 mutations, most of them disappear within a couple generations. They drift out. They don't make it into the next generation.
Well,
And
actually, because they're nearly neutral and they're not selected for or against, they would actually go along. They would drift into other populations. That actually makes the problem worse. They're not just going to be. They need to be selected out. That's the problem. They remain in the genome. There's not only a personal level, right? We die primarily due to mutation accumulation. The population as a whole. I mean, if you think about it, let's say 60 or 100 new mutations per person per generation. If there's 7 billion people on the planet today, that's 700 billion new mutations this generation alone. And you're trying to say that there's going to be no overall effect on genotype? I mean, I don't understand your logic.
There's not 700 billion mutations occurring every generation and piling up in the population. They're drifting out rapidly. They're drifting out and they're being selected out. You can't just multiply the population by the number of mutations per person and say there are 700. I mean, that sounds pretty doomsday. It isn't that bad. Relax.
Exactly and that's exactly what we see is entropic degeneration on low levels with the.
We're not seeing it
with the disease. Well, I just want to ask you a question. You did agree that these neutral mutations are unselectable. So why do you keep saying that due to genetic drift and say other evolutionary mechanisms that they are going to be selected away? No, that's the problem. Is they are unselectable. Like, are you saying because the genome is highly redundant in areas that they're just going to be absorbed, these mutations? Like, I don't think you're being specific enough. I don't understand. What's the case you can make why all of these nearly neutral mutations really have no effect? Like, is it the redundancy of the genome, or like what on a molecular level stops these nearly neutral mutations from having any type of effect?
You're aware that every single gene has its own mutation rate, right? Its own rate. Every one is different. The gene that basically mitochondrial DNA, those genes have got to function. If they don't function, that mitochondrion dies and never makes it into the gamete, right? It never makes it into the next generation, right? There are genes that have a very low mutation rate because they are vital to the organism. The ones that are mutating that you're talking about are in that big, vast junkyard. I don't like the term junk DNA. They're in the workshop, churning around, trying to find something to do. They're robots looking for a job to do, and every one of them makes a unique shape. And every now and then, like, one out of a million or one out of maybe 100,000 have some beneficial effect. And if there's selection pressure, that effect will pop out. So what I'm saying is that it is junk DNA.
Okay, so if I'll ask you
Nobody uses that term.
Right,
by
right.
the
Okay.
way.
Evolutionary leftovers, genomic fossils. I would
Not
say
leftovers. Workshop.
Okay, so I would say that the majority of mutations, okay, they fall into either nearly neutral or harmful. Even if I gave you one or two truly beneficial mutations that are not deleterious in any way,
Right.
just for sake of argument, okay. But the thing is, one or two, truly beneficial mutations is not going to counterbalance the damage. But the neutral mutations, okay, they do at least change DNA sequence, but a lot of them, due to the redundancy, they don't alter the DNA sequence. But that built-in redundancy for the code, which I'm sure you know as and is
30% roughly,
like degenerate codons and such. So that built-in redundancy is good for the organism. So even that will still have a tiny effect on genotype. And you can comment on that, but I do want to ask you, because you mentioned junk. Do you agree that based on let's say ENCODE, for example, based on studies in these areas on genome function, that the areas in the junk DNA are telling the genes when to produce their product, right, the protein, and when not to? So therefore, that should obviously be function to anybody. I mean, that sounds important. You get a mutation that is seemingly neutral in a non-coding area like that, it will still have a very subtle effect.
Now there's regulatory RNAs, small RNAs. There are all kinds of RNAs out there that are floating around, and that's what ENCODE found is all these RNAs. They 99.9% of them never leave the nucleus. Okay, they some of them have some regulatory effects, but if they have an effect that matters, the organism is going to die before it reproduces. If they have an effect that makes the organism better, then that thing is going to be copied into the next generation.
Right, basically. I agree.
There's when things go, the place where you got it wrong is you had this idea of accumulating damage, like rust on a car.
Or
what's happening. What's happening is, like, when you play 5-card poker, right? Where you draw, you get a completely new hand every generation. You're getting a new hand every generation, and if every now and then there's you're out, somebody shoots you because they thought you cheated, and then you're dead, right? Or you win, you pull 5 aces or whatever. But what in the hell? Sorry, my phone just took off.
That's okay. That's okay.
My cat stepped on my phone.
But anyway, I want you to continue what you're saying, but just before you get too off on too many different things, I do want to ask you, though, if there are truly junk areas, in your opinion, why would the cell then even bother transcribing it? Why would the cell in general even bother with it? Because it seems like that type of activity would be so wasteful. It would be such a waste of energy and resources. Wouldn't natural selection have eliminated that? If that was truly junk, natural selection should have eliminated it long ago, but obviously it hasn't. So what's your answer to that? Why is the cell bothering with all this so-called junk DNA?
The reason the cell is bothering relates to why I may not be an atheist for very long. I'm a computer programmer. I have been for 40 years. And I've looked at the code, the genetic code, and I've looked at the code in cell biology, and I've determined that it's something that we programmers call spaghetti code. Have you ever heard of that?
Mm-mm.
Spaghetti code is when you have a novice programmer, and every time they do something, they do it a different way, or they'll take the long route. They'll write 100 lines of code to modify a string when they could have done it with 1 line or whatever. It's called spaghetti code. Looking at the genome, I'm starting to believe in the spaghetti monster, because I think the spaghetti monster created it, right? So the
wait, are you saying that
you
might be believing in an intelligent designer based on the complexity of the genome, since you're computer program? Is that what? But you just not.
If you imagine an intelligent designer that's half crazy and smokes crack, and whatever, then maybe. No, there's no intelligent design, and that's the answer to your question. The reason that the cell does that is because the cell can't help but do that. It's not a perfect mechanism. The only thing that matters is that the cell survives, that the cell performs its function. Okay, it doesn't do it perfectly. There's all kinds of crazy stuff going on in there. That APOA1 melanovirus or mutation virus, they're going to find some very shortly. You're going to hit me with a paper where they find that it does have some deleterious effect, because everything works that way. It's a mess in there. Cell biology is a mess. Now, I have a theory about.
Are you saying that? Are you saying that? Let's just say hypothetically, I give you a reason why that melano is truly a loss of specificity or deleterious in some way, because it has an advantage, or because you can point to other, let's say, beneficials, whether it's, I don't know, sickle cell anemia or something. Even though that's obviously reductive, deleterious, but there's still an advantage to be had. Are you saying that there's kind of a trade-off in the genome, which would balance the?
Yes, all the time. It's always a trade-off. You're
right, but is that trade-off
cell,
sustainable though? That's the question. You know, is it?
It is. Well, not always.
Beneficials are rare. Is that trade-off?
We have extinctions. You know, there are extinct species. You know, if a species goes through some population bottleneck and starts inbreeding,
Right.
you mentioned the mammoths.
Yes.
That happened to them in two different areas. They started inbreeding. They became homozygous, and and then their mutations basically their mutations drifted through the population to the point where they went extinct. That happens. I mean, this life thing can go wrong. There's no guarantee on this. In fact, it has to, and it should, because what you got to understand about evolution is that there's only one life. There's only one living thing. All of us are the same living thing. Actually, the gamete line, it goes all the way through from the very beginning, from the first eukaryote up to us. We are all one creature. Okay. Well, actually, we're just mushrooms that pop up out of that one creature, but it's the gametic line that makes a difference. And of course, I lost where I was going with that. So I'm sorry.
Well, would you agree then that most beneficials that have been studied and looked at are reductive? There are deleterious effects.
There has to be, because one protein does so many different things in the cell that you're going to find a negative and you're going to find a positive. You're always going to find something wrong with them. But it's the what do you call it? At the end of the day, net gain, net loss. Right. That happens in every cell. A cell is just a bundle. You know, inside the cell, you kind of imagine that there's a lot of water in there. There ain't that much. It's like 50% proteins. The proteins are that close together. They're all bumping into each other. They're all mutated as hell. There's all different shapes. We mutate throughout our lives. It's a consensus. Biology is a consensus. Okay.
Well, I think it's by the time you're 40 or 50, you've accumulated like 40,000 new mutations in one cell alone. In one cell alone.
I'm 69. You can imagine how defective I am. I mean,
and think about it. If that's on a personal level, okay, if we start accumulating mutations, okay, in our somatic cell lines, at the first division of every cell division, we're accumulating new mutations. Yes, of course, it's the reproductive cell line mutations, that the ones in the germ cell that are actually passed on. The somatic cells are just pretty much the reason why we die. I mean, 40,000 new mutations in each cell by the time you're 40. But because you've agreed that, let's say, most beneficials are reductive, there's a trade-off. So we agree that there's a trade-off. My question would be, and my issue is that if you're just throwing out all this information, okay, from, say, lots of nucleotide sites, and then what you're trying to do to take evolution forward is replace all that information with, say, one single desirable point mutation, or maybe a beneficial duplication, or whatever you can point to. I don't see how that's sustainable, because we've actually seen that, and we can discuss this too, since I'm bringing it up. I don't want to take you on a rabbit trail, but Lenski's experiment, for example, we've seen a trade-off. And what that trade-off led to was a shrinking functional genome size. That's what this trade-off leads to. So I don't know how that's going to take evolution forward and take your pond scum to people. I mean,
Lenski has about 70 or 150 papers. I've only printed out like 20 of them, but I didn't find anything about it. Well, yeah, some of the genome shrunk. I mean, he had a lot of effects, but the one you're talking about, the citrate antiporter, basically, that was a beneficial mutation. It was a gene duplication, so the genome got bigger. It actually duplicated. And then, but what didn't get duplicated is the regulatory part, the part that gets shut off with oxygen, right? So what happened is, you're right about that one. It was a loss of specificity, but it was exactly what the organism needed to thrive. I mean, that happens. Loss of specificity and gain of specificity happens all the time. So, I mean, you got that part right, but it was a real mutation too, and it was an advantageous mutation. And if you let all of Lenski's bacteria go in the wild, you've said, oh, they're defective, they're not going to live in the wild. Bullshit. E. coli will survive. Those E. coli.
That specific E. coli with, shrinking functional genome sizes and, loss of specificity. I mean, because what they're doing, and, I can send you papers on it, it's pretty well known, is they're going through reductive evolution, right? They're getting rid of all these genes for adaptive purposes, but how can you not say that it's long-term degeneration? I mean, we have a shrinking functional genome size. I mean,
yeah, bacteria don't have that big junkyard that we do, or that warehouse that we do, that workshop that we do, and they can screw themselves, and some lines will draw out. But bacteria, like in Lenski's experiment,
I just want to make it clear to the audience, I don't agree that we have a junkyard. I believe that 80%
it's a workshop
being transcribed in RNA definitely suggests function, and the accumulating papers every day give us more and more inclination that the genome is highly functional. That means one single neutral mutation, which you say is absolutely neutral, it's going to have a trivial effect. I mean, I need a type of selection that's going to remove these types of mutations, because you keep saying that they just keep accumulating and accumulating and they lead to forward evolution, and selection doesn't need to select them away, but selection really does need to select away these mutations.
You already believe in it. I mean, you believe that these kinds came off of Noah's Ark, and there were 700 or 7,000 of them, and they mutated. There were beneficial selections that caused each of those animals to speciate and spread, right? You agree with that, right?
Well, would you assume, because I understand that evolutionists assume that the vast majority of DNA differences, if not all DNA differences, are the result of mutation. What do you believe we attribute the majority of DNA differences to?
Created heterozygosity.
Well, okay, so obviously you've been listening. That's good. So you'd understand that our genome is not, all those nuclear DNA differences, those tens of millions of nuclear DNA differences, aren't the result of mutation. If they're the result of created diversity, right, pre-existing functional DNA differences, well, then just processes other than mutation, like recombination, gene conversion as well, that can produce new species rapidly. So it just depends.
I can ask you the same question, basically. Okay, if you have two genes, right, side by side, and they're heterozygous, right, and they're in the population, how in the hell is that going to amount to all of the speciation? I mean, just what climbed off the ark? I mean, you've got some serious inbreeding problems going on in that Noah's Ark story. I mean, I've never gotten over how you got around that one.
No, because you talked about it earlier. Okay,
I did.
Inbreeding is the exposing of the hidden reservoir of genetic mistakes. But if these DNA differences are not the result of, see, the thing is, you need to put yourself out of the assumption, out of the evolutionary assumption, because the biggest difference between my view and your view, okay, regarding the origin of genetic diversity in general, is that I'm explaining the vast majority of nuclear DNA differences not by mutation, okay, and that's why you're suggesting that there'd be an inbreeding problem. No, because that would have to involve mutation accumulation, of course. But this origin of genetic differences that we look to, that has massive implications for the timing as to the origin of species. Because if you're trying to explain the origin of species just by mutations, yes, I totally get where you're coming from. Okay, you need millions of years for all these differences to accumulate. I completely agree with you. But if the differences are there from the start, the simple operation, as I've pointed to, of recombination and gene conversion and other such processes, mutations can provide a little variety as well. You will produce visible variety in a single generation, but you need to not strawman the position and assume that those DNA differences are the result of mutation. Now, you guys in your out of Africa theory, there would be an inbreeding problem, because now you have the 10,000 Homo sapiens inbreeding for a prolonged period of time. Now you're the one with the problem. How do you explain that one?
You mean our problem's worse than your problem with just, what, 6 coming off the ark? And like 4 of each animal, or 2 of each animal? I mean, our problem is bigger with 10,000 to 100,000. I don't get it. What do you mean? How is it bigger? We have a smaller problem.
Okay.
You got a big problem.
For the audience sake, can you explain why inbreeding is a problem in general?
If you get a real small population and you start inbreeding basically with your sisters and your cousins or first cousins, you can end up with certain mutations drifting basically into the population. You combine 2, like for hemophilia, you'll get 2 genes that are, hemophiliac, and then you got a problem. Then you've got a negative effect. And if you have enough of that, it's possible for a population to drift into extinction. But it's also possible for a population to drift into something different. Either way, you can't put a value on up and down with evolution. It could go either way. But yeah, inbreeding is kind of a problem. And I'm not. I'm just wondering about Noah's Ark.
Just one sec before we go further. So these genetic mistakes, these recessive mutations, if you're inbreeding, they're going to come to the forefront. Okay, they're going to lead to genetic damage. Of course, that's why inbreeding is not recommended. It leads to genetic damage. I mean, look at the 10,000 cheetahs. These conservationists are looking after them. The sperm is degenerate. I mean, these cheetahs can go extinct due to inbreeding. Now, my position regarding the out of Africa, because you asked why that would be a bigger problem, that's because that evolutionary bottleneck hypothesis, okay, you got Homo erectus in Africa for millions of years. And I'm sure you're aware of this, Homo erectus, there's a first out of Africa scenario where Homo erectus migrates. Then you have Homo sapiens, which evolve from Homo erectus, live in Africa, and around 70,000 years ago, I believe the evolutionists say, or the evolutionary literature says, there was an extended near extinction event that was associated with the severe inbreeding that you're referring to. So how can this prolonged bottleneck population not go extinct? And if they didn't go extinct, they would at least be genetically compromised. And you want us to believe that genetically compromised population of, say, 10,000, I've heard some evolutionists even say 2,000, seize dominion over the entire planet? That's a problem. That's bigger than the Noah's Ark problem.
I think 2 or 10,000. The thing is that you're rolling the dice. I mean, that mutation could end up it could be either one of those. Basically, either one of those alleles. Okay, right. You're rolling the dice. It's a 50-50 thing, and if you make it the right way, I mean, hell, the chromosome 2 fusion was a hell of a dice roll. Okay, and ended up taking over the population. But I mean, that happens all the time. It's happening outside in the woods right now.
Well, I don't want to go on a rabbit, and we can talk about chromosomes. We don't want to go on a rabbit trail.
We'll do that on another. Another hangout.
I am enjoying it. I want to at least say, okay, from our perspective, okay, given created heterozygosity, given our population bottlenecks, we have 2. Okay, as you know, we start with just 2 people. That's very benign. That is a benign population bottleneck because it's 1 generation. Okay. Then we have 10 generations later, a second single generation bottleneck of 8 people. This was at the time of Noah that we're talking about here. Both bottlenecks, just like Adam and Eve, were very brief, just 1 generation, then followed by massive growth. Okay. In both cases, there would be almost no previously accumulated mutations. That's why there's no
Why?
inbreeding problem with us, because there's no accumulating mutations. But with you guys, just real quick, and I'll let you talk as much as you want. For us, this limited human genetic diversity is not a problem for us. It makes sense coming from two people, Adam and Eve, but it's a huge problem for you because your evolutionary bottleneck hypothesis involved an extended near extinction event associated with severe inbreeding. I mean, you even told me that you explained the majority of DNA differences as mutations. So how is this even remotely feasible, this out of Africa scenario? I mean, there would be such permanent and severe genetic damage. These enormous numbers of deleterious mutations would go to fixation. You talk about fixation all the time. They're going to fixation. So this tiny, nearly extinct, genetically compromised population, as I've said, suddenly explodes into all parts of the world, seizing dominion over the world, and here we are today. I mean, can you convince me as to how that's actually feasible and not just post hoc or ad hoc?
Okay, you're let's see. First of all, Noah's Ark. Okay, that was 1,600 years after Adam and Eve, right?
Correct.
So they must have had a bunch of mutations, and there are only 8 of them. And you're telling me that there were no mutations? I mean, the fall happened right in the first week, didn't it? When they ate the apple. So, I mean, mutations were all over the place for 1,400 years, and then we only have 8 people. That's a bottleneck, and that's a problem. But that's not a problem for
do you believe
survival. Well, I mean, there are species that
50 to 100 new mutations back then. Okay, if we have a genome of 6 billion letters, 3 billion from Ma, 3 billion from Pa, 1,600 years of 60 to 100 new mutations per person per generation is not going to be nearly as bad, especially because we explain the vast majority of DNA differences are created differences, as your situation in the out of Africa model. I mean, you need to explain how that would be a problem, because according to our model, most mutations came after the flood anyways. Environmental conditions, post-flood radiation. That's why you see such a significant decline in age. From 900 down to 100 down to today, the average lifespan's what?
You thought the
80 or something?
You thought the radiation rate was a lot higher before the flood, I thought.
No, because before the flood, there'd be less mutation accumulation, but post-flood environmental changes would lead to more mutations, because a lot of mutations are environmental. Are you familiar with your out of Africa theory, though, regarding because from my understanding, the way the evolutionist explains the low genetic diversity that we see today is a population bottleneck, right?
Yeah.
The population of Homo sapiens 70,000 years ago was reduced to between 2 and 10,000. And then that's how they explain the evidence for mitochondrial leave and Y chromosome Adam is one thing.
I think it might have even been smaller than that. But let me tell you about Blatella germanica, okay, the common cockroach. Okay, have you heard about this guy?
What's the name?
Blatella germanica. It's a cockroach, the common cockroach.
Right. Okay. Yeah.
Pyrothins kill them, basically. Okay, and there was a mutation that occurred, and I can explain the mutation to you. It has to do with the voltage-gated sodium ion channel in the neurons, and it's what killed them. It's why pyrothins kill them. There was a very, very slight deleterious mutation that changed the shape of that protein, or that changed the shape of that protein channel, so that pyrothins no longer attached to them, and the cockroach could keep living even though they sprayed them with insecticide, all of them. Just a few cockroaches popped out, like one with this mutation, and somehow he spread so that it became a real problem. Okay. Now the mutation was slightly deleterious in that the sodium channel did not work as well, but then 2 other mutations came along from somebody else, created heterozygosity, and made it work well again. So anyway, so now all of a sudden we have a insecticide resistant cockroach, which is a problem. And they've studied it very, very thoroughly. But these bottlenecks can result in extinction, or they can result in speciation, one or the other. It happens all the time. It's happening out there everywhere in the woods, right around us. We don't know all about all of them. We haven't studied all of them because we are crap at. We're slow as humans. It's going to take us a while to go out there and figure out what's going on. But you're seeing population bottlenecks all the time that result in a speciation event or just a species that survives.
Well, depending on the length of it.
It doesn't matter.
No,
it's a roll of the dice.
But of course, the length of the bottleneck would matter. Like, for example, if it's 2 to 10,000 Homo sapiens, okay, that have a prolonged many generations bottleneck, that's generations of inbreeding, because we can see it in the 10,000 cheetahs in the world today. I mean, there's just not enough of them. We can see that the genetic diversity has eroded. It's due to inbreeding, and the species are starting to express many destructive recessive mutations. So I just don't understand how modern Homo sapiens 70,000 years ago, who would have obviously been genetically compromised. I understand your example there, and that's fine, but is it really any more than just post hoc, ad hoc evolutionary storytelling, right? Like, why should we believe?
Wait a minute. The bottleneck has been around for a long time. It's not ad hoc, not post hoc. We're not making shit up. I mean, the bottleneck has been verified with a lot of different information. I don't like when you do that. That is disingenuous, basically.
Well, how is it verified?
We don't make shit up. We try to figure out how something worked.
Well, what's the evidence for the bottleneck 70,000 years ago?
The genetics. Basically.
Right, exactly. So evolutionists didn't predict such low genetic diversity. I mean, me over here in North America, well, you'd have to provide me a paper that says, hey, we predict low genetic diversity before we actually discovered it. And then, okay, maybe I'll admit to that. But according to the Bible, we started with just 2 people, Adam and Eve. Low genetic diversity is easy to explain. It's expected. It's predicted.
Yeah, but now, you guys didn't even talk about genetics before 10 years ago. I mean, you guys just suddenly became scientists. Before, like when I grew up, creationists didn't believe in that. They didn't believe in biochemical life. They thought there was a life force in the 50s and the 60s. So don't give me that you guys beat us to this prediction.
Okay, well,
this prediction made sense.
Well, here, I want to ask you a quick question that I asked in the opening. If there really was Adam and Eve, okay, we started from 2 people, okay, what would you expect to find in our genetics? You being an open-minded, skeptical individual, what would you expect to find?
Incest. I don't know.
Okay.
I think it's possible. I think it's possible for any 2 individuals from species to create, to basically sustain that species and to multiply. I don't see, with the right dice rolling, I mean, you can basically marry your sister and have a perfectly fine kid. So, which obviously happened, right, according to your story. But I don't see a problem with that.
Because once again,
I don't know what would happen.
Yeah, but I have to point out your assumption for the audience. You're assuming that we explain the vast majority of DNA differences as mutation. When we accumulate 100 new mutations per person per generation, we have a lot of other inherited DNA differences that are being passed on that are not mutation related. So even the evolutionist actually resorts to and looks to pre-existing heterozygosity. Yes. We're just saying the original heterozygosity was engineered by God. That's also why we look to function, of course. But the question I asked you, you didn't really answer it honestly, I find, because I think you knew in your head, you probably thought, okay, I would expect to find low genetic diversity, evidence for for a Y chromosomal ancestor, evidence for a mitochondrial DNA ancestor. And we can look at observed mutation rates, not accounting for evolutionary assumptions, and it takes us right back to 6,000 years. I mean, everybody's Y chromosome and everybody's mitochondrial DNA is incredibly similar. That's why the evolutionist looks to the population bottleneck. Even they believe in mitochondrial, even Y chromosome, Adam. Of course, they resort to unrealistic mutation rates and phylogenetic assumptions to come up with their date as to when they existed. But that is the evidence that one should expect to find if they're open-minded and a critical thinker like yourself. I mean, what other evidence would you want to find for me to convince you that, hey, listen, Adam and Eve exist. The Bible is true.
You're not going to find it in the genome. this. Oh, where the hell was I going with that? Totally lost my train. It's really late for me. What were you saying again?
No problem. I want to say that actually this was a lot of fun. This is a really
Okay, Adam and Eve. What do
I
you
wanna do,
expect?
since you're the guest, I'm going to give you a few final thoughts.
Okay.
We can do another discussion on this too. We can both maybe go look at some of the things that were talked
Yeah,
about.
we got a
maybe go
bunch of
little more-
questions too. So
Okay. We should do it again. We should talk about chromosome 2 next and a
Yes,
bunch
I
of other.
agree.
But there's a really much bigger discussion, and you keep asking, how does a tetrapod limb form, or that sort of thing, and there we get into morphogenesis, which is something that I know quite a bit about. And in one of your videos, you said we don't know anything about embryology and morphogenesis. I have stacks of books on it. We know a lot. Okay. And that's where evolution happens. And that was the most profound thing that happened to me is when I started studying embryology and morphogenesis.
Well, I never said that we don't know anything. I was just saying that I was saying on a molecular level, we know very little about how the organism from a single-celled organism, from an embryo, a zygote.
We know a lot.
Hey, are you saying that because we really are just beginning to understand and comprehend this developmental process, how coding and non-coding DNA coordinate, when, where, and how these structures are sequentially put into place to produce a fully functional organism? I was talking about on a DNA level, how this happens, step by step. Are you saying we understand all of that perfectly?
That's what my books are about. How it happens, we can talk about that. And there's a really, really exciting thing about how it happens. It doesn't take a whole lot of genes in order to make a genetic change in morphology in an organism, in order to turn a limb into a fin or whatever. But I am very tired. We'll get into that another time. And you say there are questions. Do I have to endure questions from your crowd?
Praise, what we'll do, because we're going to start shutting it down. We did, I don't want to keep Speed of Sound here too long. We did agree to an hour. It's late for him, especially he's been up since 4 a.m. So we're definitely going to continue this discussion. But that being said, is there one or two questions, Praise, that you're just dying for either Mike or myself to answer?
Yeah, how about 2 questions? How about that?
Sure.
Well, first you had a super chat from Coco Puffer, and they said, "Thanks SFT and crew for your work." So that's pretty cool to get that. And
thank me.
Thanks. Of course, if there's any super chats, always put those to the front. We got to read those off. We just got actually, so that's cool.
They forgot to thank me.
Yeah.
I'll thank you, brother. Thanks for coming on and doing this discussion.
Yeah, great discussion. So Nephilim Free, this is from Nephilim Free. So his question is, since the majority of SNPs cause negative effects to protein functionality, why are we to believe mutation could design proteins?
We would have to have a long talk about protein folding, basically, and I look forward to that discussion too. I think we should do it another time. But there are every protein folds, and there are a number of different shapes, and we have to talk about the sequence space, and there's some mathematics and all kinds of stuff to consider there. But it would take me another hour to begin to answer that question. So there are reasons for it, things I believe in. So Nephilim, I appreciated our talk the other day, and I will try to address that in the future. Okay.
Cool. So here's a comment from Raw Matt. He says the Human Genetic Mutation Database, HGMD, over 3 months, more than 6,000 new harmful genetic mutations are registered, which is an annual growth of over 24,000. So if you can respond to that if you want.
Yeah, we're getting really good at finding things, basically. And the more we look, the more we find. And of course, the more are created. And there are all kinds of, I'd love to see that database because, like, have you ever gone to a playground and looked at 50 kids out there on the monkey bars? Why do they call them monkey bars if we didn't evolve from monkeys? Anyway, you look at these children, they're all healthy, millions of them. We are healthy. I mean, overall, we are healthy through our 20s, which is all we need for evolution, basically. I don't.
How much of the genome, because I know you talked about junk law, I'm just curious, I know we're in the question portion, but how much of the genome, considering non-coding and protein coding, would you say on a percentage level is truly functional, right? Functions in whether it's embryonic development, the timings of protein manufacture, regulatory functions. I mean, how much of it would you say?
With us, I'm guessing about 12%. I may move that up to about 20. But I think we have 80% slack out there. But you got to be careful with function, because some of those long ALUs, these long stretches of DNA, they're important for cell biology, because if you take a string and you have to hook it from the doorknob to another doorknob, it has to be a certain length. So I mean, there's that kind of importance, because in a cell, DNA is structured, very, very highly structured, and that's how it gets. That's how tissue differentiation happens. So anyway, it's but I'd say 12%. I think is kind of a guess. The rest does something, but it's not.
If according to our model of pre-existing heterozygosity, right? Because if God encoded Adam and Eve and the created kinds with these functional DNA differences, and we are therefore predicting that the vast majority of these DNA differences should be functional, therefore we are providing a novel future testable prediction that, say, based on comprehensive knockout experiments, we know ENCODE only studied about 147 cell types, and then considering the human body has what, a few thousand cell types, we are expecting more and more function to be found. So we are saying that the vast majority, probably over 90% of the genome, is functional. And since you just said that you believe it's about 12%, considering some of the other things that you talked about, maybe 15 to 16% if you consider that, wouldn't that be a future novel testable prediction coming from our side that only future observations and future tests can prove to be either true or false?
That would give us pause, but it depends on what you mean by functional. And like we'll talk about the chromosome 2, the bridging protein, but anyway, it would give us pause. I would not concede evolution because of it. I wouldn't be surprised if you could find some small function for all of those little pieces of mRNA. They all do something. They're all molecules. This is chemistry. It's nothing but. I mean, if you have chemicals in your cell, they're going to do something, okay? Whether or not they're vital is the important part. So you don't want to talk about deleterious mutations. You want to talk about vital, mutations, that sort of thing.
You would have to agree that if what we're saying is true, okay, so if
it'll give us pause.
Right. Well, and also, it would show that our thesis of genetic degeneration, okay, the fact that you're saying that most mutations are neutral and there's no harm at all to genotype and phenotype is wrong. Because if 90% or more of the genome is functional to some extent, that means these neutral mutations are nearly neutral. That means they are degenerating our information systems. This is why evolutionists, there was a paper that came out recently where the genome, like you said, it can't be any more than 15 or 20% functional, or else man and chimp, both lineages, would have degenerated. Evolution can't be true. I mean, they have to keep most of the genome junk, right? So that's a head-to-head prediction. Because if we're saying, we need to look at some knockout experiments, we can't just look at biochemical. That's what ENCODE tested for, okay? So it is preliminary, but it does set a trajectory. That's why we made predictions. That is a head-to-head test and prediction that both the evolutionist side and the creationist side can do. And I'm
Are you predicting that 90% of the genome will be vitally functional? Vitally. If we knock them out, the thing dies.
It depends.
Is that your prediction?
That's a good question because a lot of it, because they have knocked out genes in some mammals like mice, but some RNA genes or some non-coding genes DNA may only be functional or expressed in certain developmental pathways. Right? So just because you snip out a section of DNA, well, you know what, that might be redundant, playing a purpose there. I would say purposeful, functional, and overall vital. Yeah, I mean, if I were to remove something in my car, There's a lot of parts to the car. So even though it is important to the car, it's functional, it's purposeful, the car may still run. But yeah, over 90% functional, purposeful. Vital's a tough way because you know what? Some of these genes, although functional, doesn't mean the organism's going to die, right? Just because it's knocked
but
out.
valid in some environment that God has predicted. If you find that 90% of them are vitally functional, I'll eat one of my cats. I got 2 of them, so don't worry.
Then you're only left with one. Okay, and that helps me understand the position.
Yeah.
Praise. Was there any more questions, or are we going to wrap it up?
Yeah, there's several questions, but I don't know if Speed wants to go through all those. But I will say one thing before we get going: that Neff is holding a hangout. If people want to join into that after this, and just send an email, and you can go into that and join.
Oh, you guys are going to roast me.
Hey, we're nice guys. You don't got.
I'm not going to be there. Yeah, you're nice guys. I've seen some of your nice guy videos. Anyway, okay. This has been fun. We should talk more because there's a lot more to talk about.
Yeah,
there is.
And you'll learn. I hope you learned stuff tonight. I certainly learned something about your position. I would like to learn more about this whole Noah thing and how those 8 people made it, all of that. But anyway, it's been fun. Thank you.
Awesome. It's been a blast. Thanks for coming on. You were a guest. I hope you felt like you got enough time to talk. You weren't interrupted. It was respectful. And we'll definitely do it again sometimes. Very enjoyable discussion. So Praise, thanks for setting it up, and we'll go from there.
Hang on a second.
Have the same accent. That's pretty cool.
Oh, you hung it up? I just want to say that I'm pleasantly surprised at how polite you guys are. And how good you've been. I thought it was going to be a different situation. So anyway, thank you.
All right, man. Glad it was okay for you. So we're going to end it here. You want to say anything before you end it here, SFT?
No. Like I said, well, I think capping on what he talked about, sometimes in the comment sections, right? You're looking at a text, may come across a little more heated, or there may be a sense of animosity there. But no, once we get in here and we're just 2 human beings talking about differences and worldviews, there's no reason why it can't be respectful and therefore profitable. So I appreciate that, Mike.
Yeah, that's what I found out when I hung out with Nephilim. I really like that guy. And I didn't think I was going. I was trying not to.
Yeah.
But I like them. So see you guys.
All right, guys. See ya. And this will be the end of the stream. Later, everyone in the chat, go to Neff's room.